Hello, my name is Florian. Thank you very much for tuning into my presentation entitled Machine Learning Guides Identification of Virus Antigen Specificity based on Deep T-cell Phenotypic Profiles. I'm here representing Immunoscape. We are a company that is working on the development of novel TCR-based therapeutics against solid tumors. So let me start the presentation telling you a little bit about the biological background of TCRs, of T cells, and why this is an interesting field. T cell receptor, or short TCR, is a surface protein that's anchored in the surface of T cells, and it's used to detect antigens, which are short amino acid sequences coming from larger peptides, and those are presented by antigen-presenting cells, usually on MHC class 1 or 2 molecules. You can see this in a schematic representation here on the left, and in 3D here on the right. And essentially, the detection of antigens is necessary for T cells to start acting on their immune function. Next, the TCR is the key element in deciding which antigen a T cell can bind to. There are quite a lot of TCRs in any human being. There's the estimate that there are about 5 million unique TCRs in any healthy human person. And if you just do the mathematical experiment, thinking about it, there are 8 billion people around the world, and we're estimating that those share about 11% of TCRs, then there will be a pool of 3.6 times 10 to the power of 15 unique TCR sequences. So this is a really huge number, representing the entire spectrum of T cell receptors. And now, considering this huge number of possible T cell receptors that can exist, there is the obvious question to predict which antigen each of these TCRs will bind to. And this is actually a very important problem, not just to better understand autoimmune conditions, but also there are many applications in oncology for new drug developments, for enhanced TCRs, for cancer vaccines. It's important to understand the relationship between phenotype and T cell antigen specificity. We will dive a bit deeper into this particular point during the presentation. It's also important to learn about cross-reactivity predictions in T cell therapies. Predicting which antigens would bind can help in the design of new therapies and vaccines, also out of oncology. And it's also really important to better understand the role of T cells in surveillance of malignancy. So even though this seems to be a very interesting and important problem, it's very hard to solve for many, many reasons. And some are summarized here on the slides. So one very obvious problem is limited data. And this shows up in different ways. So one is that there's a bias in publicly available data towards the beta chain of the TCR. So earlier you saw that there were the alpha and the beta chain, and most public datasets only contain information on the beta chain itself, which is not sufficient to really model the binding of the TCR to the antigen. Also, the HLA distribution is highly unbalanced, so we do not really cover all HLAs properly. And this also relates to another problem, which is that for certain antigens, we only have very few TCRs, mostly just one. So you can see that for about 25% of antigens, we only know one TCR that actually binds to it, which is not sufficient to really describe the space that this particular antigen could be bound to. Another problem is that it's very complicated to obtain negative data, so it's very hard to know which TCR guaranteed does not bind and does not cause a functional reaction to a certain antigen. It's also very challenging to incorporate structural data. This is complex, mostly also because there are just not enough structures available. And because of all these reasons, when we look at state-of-the-art models that try to predict peptide MHC interactions, then we see that most of these models perform very badly on unseen epitopes, so epitopes not used for training. So mostly the performance there is very close to random. So because of these challenges, we thought to simplify the problem in a divide and conquer manner, if you want to say so. So we tried to propose that instead of predicting the TCR antigen pairs, maybe we should just first try to predict the virus specificity of a certain TCR. So in this way, we can simplify the problem because instead of looking at the entire space of epitopes, maybe we can just look at the space of CMV epitopes or maybe the space of EBV epitopes. This should make the downstream problem of figuring out what's the correct antigen a lot easier. So let me explain you the study design and overview on the experimental method. So every analysis starts with our targetscape assay. Targetscape is a highly multiplex staining approach where we take PBMCs from a patient and then we stain them with hundreds of epitopes at the same time. And this multiplexing using different metals allows us to really scan for all of those simultaneously. And what we get from this using CyTOF machine in the end is um, information on T cell specificity for many T cells at the same time, and also information on a set of surface markers that helps us to phenotypically do an initial characterization of all T cells that are specific to a certain epitope. And using this targetscape assay, we have profiled PBMCs from 169 samples, including 114 healthy donors, 55 cancer donors, and within those 169 samples, we looked at T cells specific for CMV, EBV, flu, and COVID. We looked at 32 epitopes in total across various HLA types, and we have profiled 25 surface markers and 2 intracellular markers within those virus-specific T cells. And this slide provides you an overview on the different T cell populations we found that were specific for CMV, EBV, flu, and COVID across the different HLA types. And it really shows you that we are quite broad, so it's not the usual AO2 bias. We really cover many HLAs across the different viral categories. And before doing any machine learning on this data, we looked at it using traditional approaches, here just having a heat map on all phenotypic data that we obtain. And we can see nice clusters of virus-specific cells. For instance, we have CMV cells on the left side of the slide here. We have EBV here on the right. We also have a group of MART1 specific cells here in the center. And we have 2 groups of EBV cells, the blue group here as well as the blue group here. Interesting enough, the COVID-specific cells, they do not form a unique cluster. They are really more diverse. You can see them in different parts of the heat map. And this also fits to related studies that were published recently, showing that the phenotype of COVID-specific cells is rather diverse. What is nice as well is that when we translate this heat map essentially to a UMAP, we see a similar clustering, or I should say grouping of cells, where EBV cells form one group, the CMV cells form a group. COVID is slightly more spread, flu is also very nicely specific, such as the MART1 cells are as well. So now using this phenotypic data, which seems to contain a lot of signal on the specificity of T cells, we will try to build a simple logistic regression model using lasso regularization to actually predict virus specificity. And the way the data looks like, it goes into the model as shown here. So we have a model schematic where we have all our cells with the different features, which are our 25 surface markers and the 2 intracellular markers. Then we try to fit the model such that we get the regression coefficients that will tell us in the end about the virus specificity of each individual cell. And model training and testing is done using 10-fold Monte Carlo cross validation, where we split our data into 80% training and 20% test. And for the training, we also have a 6-fold inner cross validation, and we also perform data sampling such that we do not have to deal with unbalanced data during model training. And here on the slide, you see the model performance in terms of balanced accuracy and also F1 score for test error in blue and training error in orange. And you can see that overall the models don't seem to strongly overfit. it seems well. What we see is that we do obtain very good models for EBV. CMV models seem to struggle a lot more in predicting CMV stats accurately, and flu shows intermediate performance. We can also go ahead and look at the regression coefficient. So this is also why we use a linear model, so it's easy for us to interpret it. But it's very obvious here that CD45RO is a very striking marker for EBV. We have also similarly strong markers for flu and CMV, but really the CD45RO is the most striking, strongest signal that really suggests that CD45RO is a good marker for EBV specificity. And what we can then do is we can just go ahead and check if indeed those top markers predicted for each class are also really very highly expressed across all single cells that we have. And indeed, we see that granzyme B is most abundant in CMV cells, EBV most abundant in CD45RO most abundant in EBV, and CD127 is the most abundant in flu. Another important observation here to make is that model, the Lasso model, did not exclude any of the 27 features. So meaning that all of those 27 features seem to have been carrying information to actually make the predictions better. So this means in a way that our model is still sort of underdetermined. We have to find a way to add more features, more information into the model to get even better predictions. And to do this, we now follow up with our second part of our deep immunomics platform, which is single-cell VDJ site-seq. And in this experiment, what we do is that for samples for which we know from the TargetScape assay which epitopes they contain, we will perform a deeper profiling using antibodies to fish out those particular T cells again that would be specific for a certain epitope. And on those, we will perform single-cell RNA-seq. We will look at the larger pool of surface markers. We will perform VDJ sequencing. And again, we will know about the specificity because we target those cells with antibodies. And let me show you now what we do with the single-cell VDJ site-seq data. So what we have done here is we obtained the single-cell VDJ site-seq information for two cohorts, a discovery cohort of 48 samples and a validation cohort of 10 samples. And here we looked at 22 epitopes across just 3 categories, which is CMV, EBV, flu. We dropped COVID here simply because the COVID profiles were rather erratic, which might also be because we have to deal with pools of people that just got COVID infected in a natural way. Back then, there was still vaccinations going on. So it's a very vast field of different phenotypes that we might have to deal with. So to simplify the problem a bit, we excluded COVID here. And then for those 58 samples in total, we obtained antibody-based phenotyping for 138 surface markers. We obtained single-cell RNA sequencing. And for the validation cohort, we also obtained TCR sequencing data. So now the model schematic is slightly different, but only slightly. Now we have our 138 surface markers here. So the model can pick from a lot higher number of features. And now instead of Lasso, we use elastic net regularization to retain correlated features in the model, which should help us to obtain good performance. And again, we do the 10-fold Monte Carlo cross validation. And we also again do sampling such that we do not have to deal with unbalanced training data sets. And again, here we have performance measurements, balance accuracy, and F1 score. And of course, the important question is, are we doing better, having more features? So let me show you the comparison. Here we have balance accuracy on the left, F1 on the right, test error on the top row, and training error in the bottom row, where the green box plots show you the single-cell VDJ site-seq data, and yellow is TargetScape. And what we can see is that we do perform better, having more features. Even on EBV, which was already performing pretty good, we still get an improvement. Importantly, we get a substantial improvement in predicting CMV correctly. So there we do really much better now in both measurements, which really shows that if you cover more phenotypic information, you really can get much better predictions out of your models indeed. So again, we have of course also looked at the phenotypic information in terms of importance of the regression coefficients. And here we see, similar to before, the CD5RO is a strikingly important feature for EBV. The CMV features have changed very much compared to the previous model. For flu, it's also quite similar. We still see, for instance, CD127 being important. And again, just as a sanity check, we also see that the top features are the ones most abundantly occurring on the surfaces of the respective single cell populations. So to make this a little bit more robust statistically, we have then looked at pseudobug data for each donor and looked at the top features for some of the top features for CBV, EBV, and flu models across both cohorts, our discovery cohort and the validation cohort. And we do see that those top features are robustly, significantly, differentially expressed even in pseudobug space between the discovery cohort and also the validation cohort, which you see on the bottom. Another important point to make is that the signatures that the models predict actually are present across different cell types. So this shows you cell types of the discovery cohort with different canonical markers. And you see the model markers on the y-axis for the different viral categories. And you can see that in those dot plots that the dots are quite colorful, even though we go across cell types, meaning that even though, for instance, for CMV, it's known that CMV is enriched in TEMRA cells, we see the same signature also abundant in effector memory cells. For EBV, it's the opposite. Effector memory cells are more abundant, but we also see CD45RO expressed, for instance, in central memory. And similarly for flu, where the signature is not just in central memory, but also effector memory and in TEMRA cells. And this also translates to applying the model into our validation cohort, where we have used the model to recall experimentally validated cells that were CMV, EBV, or flu specific. And what the model does is that it's able to predict cells correctly across cell type borders. So for CMV, for instance, we are able to pick up effector memory and central memory cells. For EBV, we have picked up also central memory and TEMRA cells, and for flu, not just central memory, but also effector memory and TEMRA. And this really shows that the signatures that we find are not biased towards just one particular epitope. And yeah, the model also performs well on the validation cohort. So this is why we do have the validation cohort. You can see that we are doing very well on the diagonals for all the different donors. In fairness, we do struggle slightly in predicting the EBV cells correctly from HD2, which is also why we went ahead to do more wet lab validation for some of those EBV cells. And I want to show you one example here. So this is the HD10 cohort, the validation cohort, where we have highlighted one particular clonotype that was predicted to be EBV specific. And for this particular clonotype, we did validation. So we transduced TCR into reporter cell lines. We exposed this to several EBV peptide pools. And we found that indeed, towards these three epitopes, this TCR was showing reactivity. So it is indeed a EBV specific TCR sequence, showing that the model predictions actually do work. So with this, I'd like to summarize already. I have showed you today that we have generated a pretty comprehensive atlas of viral specificity across 32 epitopes and various HLAs. For a subset of those, we have performed a deeper phenotypic profiling using 100+ surface markers, single RNA-seq, and TCR sequencing. And we used all this data to train and compare machine learning models. And one of the predicted EBV TCRs we have also validated to show that the model actually makes accurate predictions. So with this, I'd like to thank a number of people that helped us with reagent preparation, sample processing, running the site of experiments, also all the blood processing, carrying out the single-cell RNA-seq experiments, as well as data analysis. And I thank you for your attention. And if you have any questions to the talk, please find me at the conference or please send me an email. Thank you very much for watching this. Enjoy the conference. Bye bye.